Honorable Shaw laureates, distinguished scientists, forum participants, ladies and gentlemen, good morning. Welcome to Day 2 Plenary Session at the Hong Kong Laureate Forum 2025. This is Patricia Young, and it's my pleasure hosting for you this morning. On behalf of the HKLF, a very warm welcome to all of you joining us in person, and to those of you tuning in via our live webinar. Yesterday, we had a grand opening ceremony and masterclasses on 2 disciplines. I believe those who had attended were excited with the inspirational and intellectual idea exchanges. Today, we're truly honored to have 3 renowned scientists in astronomy and life science and medicine to share with us their knowledge and research experience. This morning, we're privileged to begin with a keynote speech by Professor Reinhard Genzel on massive black holes, experimental evidence, and cosmic evolution. Following that, we'll move into a thought-provoking panel discussion with 2 distinguished scientists, Professor Kazutoshi Mori and Professor Peter Walter. And I'm sure many of you are familiar with Professor Reinhard Genzel. Please allow me to provide a brief introduction. Professor Genzel was awarded a Shaw Prize in the discipline of astronomy in 2008. In recognition of his outstanding contribution in demonstrating that the Milky Way contains a supermassive black hole at its core. And among a lot of other globally renowned prizes he received, he's a Nobel Prize winner in physics in 2020 for his discovery of a supermassive compact object at the center of our galaxy. He's currently the director and scientific member, Max Planck Institute for Extraterrestrial Physics, and the chair of the board of adjudicators of the Shaw Prize. Now, ladies and gentlemen, please put your hands together to welcome Professor Reinhard Genzel.
Good morning. Is it too early? It's too early for me. I go back to bed. Plus these black holes, who cares? Yeah, well, I mean, that's a question actually which many people might ask. Why am I giving this talk? You know, every movie producer knows that black holes exist. So why do we need to make a scientific subject of it? Well, of course, you know, we have these beautiful pictures from the movies and so forth. So there it is. And we know the black hole is supposed to be something black in the center. And then the light emission outside. And we know, we've heard that it's pretty difficult to get out of there. Getting in is easy, but then once you get in, ooh. Okay, well, but I mean the scientific issue, of course, if you listen to any of the many astronomy talks these days, you will encounter black holes as one element quite often, just like exoplanets maybe, or galaxies. So the question physicists, and I consider myself in that sense a physicist, is fine that we think we have these objects where gravity is so strong that light cannot escape, but do they really exist in the sense of physics? So that's what I'm trying to tell you about. Now, if you have listened very carefully, the Nobel Committee wasn't very cautious on this, right? It said, of a very massive object, probably a black hole. So that tells you that the Nobel Committee still leaves some holes open, and we are working extremely hard to close these holes. I would bet you in about 40 years we will really know, but we're not done yet. So let me tell you where we are. Now, of course, as a little background, just for those of you who have not encountered black holes, what are they? Well, mass, we know. We know the mass of the Earth, and we know what gravity is. If I try to jump, despite my bad knee, I will come back down. Every one of you will come back down. You can try it. Now, of course, the faster you jump, the higher you might get. And the physics of Newton tells us that if you take a rocket on the surface of the sun and let it go off the surface at a speed of 600 kilometers per second, roughly, then it will escape. It will escape into distant space. So that's the escape velocity, and that tells you something about the strength of gravity. And the gravity is determined by the mass of the sun, but also its size. So if we go in mind to a different sun, which has the same mass, but is smaller, then we know from physics, gravity is stronger. So if you calculate, according to Newton, the escape speed of that rocket, if it's a 3-kilometer diameter of the mass of the sun, oops, a number comes out which some of you may know, 300,000 kilometers per second, that's the speed of light. And we have very, very firm, absolutely clear evidence since the late 19th century from experiments that there's nothing which can go faster than speed of light, independent of what people tell you. That's all fake news. So, if that is the situation and such an object exists, that means that inside of a characteristic radius given by the mass and the size, light cannot exist, cannot escape. Also, masses cannot escape. Now, here's a second aspect which immediately you've learned. I talked about light as if it's particles. And indeed, during the early part of the 20th century, Max Planck and others learned about quanta and learned that photons, particles of light, have an energy, and the energy is equivalent to a mass. E equals mc squared. You may know that equation. So that means once you have such an object with this radius, then neither light nor any particle with a mass can escape. But if you bring it inside, it will fall to the center. And mathematically, that center is a point. So it's a center with infinite density. That's what the theory of general relativity by these men has predicted. A point with infinite density, and all of the energy, mass energy, is contained in this. Now, it took a long time after the publication, so to speak, of Albert Einstein of the theory to be really tested out mathematically in more detail. There is a man named Schwarzschild on the top here who asked the question, well, let's take actually a likely configuration of such a mass, namely a sphere. How would it look then? Others asked the question, what if this thing has a spin, it rotates, what happens then? What if it has a charge? So all of this got investigated in the 50 or 60 years after Einstein's prediction in Berlin 109 years ago. So it took, about 110 years before we can actually talk about experiments. Amazing. Although gravity is very well known to us all. But to learn about gravity is extremely difficult, and that you will see. Now, how can we see gravity if it doesn't emit light? Well, in principle, you do the same as in the solar system. If you had such an object we call a black hole in the center, and you had a bunch of objects with the same mass as the planets around the object, if it has the mass of the sun, well, then they wouldn't move around to first order like planets around the sun. And so that is a way to experimentally infer that there is a central mass. And you can even measure its mass, because the exact speed of the orbital motion, remember the Kepler laws for the solar system, tell us how much mass there is in the center. So that's one, obvious way to perhaps, find a black hole. Now, there are other ways, of course, and I will briefly touch on that to do this. But it took 60 years after Einstein before the first real evidence for possible existence of such black holes came to the fore. One was discovery of very bright binaries, one of which was an X-ray star. And then the other one is this one here. Objects which showed up in the radio, were measured in the radio, and then when you look up in the optical, which you see up on the top left, it looks like a star. You see these diffraction spikes. Actually a fairly faint star in this particular case, 16th magnitude, not very bright at all. You can't see that with your eye, but with a big telescope you can. And then and that was done by Martin Schmidt, the man on the left down there, you take an optical spectrum, so you disperse the light, and you see the different colors. You see the very, very, very clear-cut lines in that spectrum. And those lines have ratios of wavelength, which we know very well. And they point to hydrogen, and helium, and neon, and so forth. But they are all redshifted by 16%. Now that is pretty incredible. 16% of the speed of light. So what does that mean? Now the interpretation of the theorists who thought about that at the time, after about 8-10 years of discussion, went towards the expansion of the universe, which was already well known since the 1930s. So this is now 1960, and since 1930, we know the universe is expanding, and in an expanding universe, the further something is away and sends light to us, the more this is redshifted. So if you take that redshift they measured here of 16% and calculate how far away it would be, you come up with a number of 2.4 billion light years. 2.4 billion years of light to get from there to here. That, at the time, sounded outrageous. The universe, which was known then, was millions of light years, not billions. Now, of course, nowadays we know that the universe is 13.7 billion light years old. So this is far away, this object. Now, if it's that far away, then of course what looks like a mega faint star is in reality extremely powerful. So if you can calculate that, then it comes out that this little light point there is 1,000 times more luminous than the entire Milky Way. 1,000 times. And all of this coming from a region which is a mere light year or less across. How can that be? Now, the theorists at the time thought about this for quite a while, and then came up with what you might say is an absurd explanation, which is accreting black holes. Oh, you say, that can't be. You told me that light cannot escape from black holes, right? And now you want to tell me that this produces light? How can that be? Well, because I told you that light cannot escape within the event horizon, but outside it can. So if you let a lot of material fall onto a black hole, then you produce a lot of energy, and that energy then can produce light. So that's the proposal. That was the proposal of famous men like Donald Lyndon Bell and Martin Rees in Cambridge, and many others, but this is not proof. The proof, of course, is can we actually see that in reality? Now, to do this, we need experiments, and I'll show you 3 experiments which have been participating in the last 50 years to make the case pretty good. One is motions of stars around the center of our Milky Way. The center of our Milky Way is the nearest galaxy, so while quasars are so far away, our Milky Way is a mere 27,000 light years away, so that's around the corner, so to speak. So that you will see was done and how it was done. The next experiment in the end was finished, or at least came to successful results at about the same time as the first experiment, and that's gravitational waves. If you have masses which move very fast, well, then it generates changes in the space-time curvature, and that you can sense through gravitational waves. And then finally, you can actually go ahead and use what is known since a long time, a radio interferometry, where you take radio telescopes over the entire globe and measure the light which comes from a distant point and interfere the light. I'll say a little more about this later. Interfere the light, not just detect it, but interfere it, and thereby generate what is the size of a telescope equivalent to the size of the Earth. And that gives you enormous resolution. And that was also a very important part of the success of the last few years. So let's say a little bit more about the technology. Now, I won't go into too much detail, but of course the first thing is we need resolution. These black holes are very, very small. If you take the mass of the Earth as an example, then the event horizon size of the Earth would be 1 centimeter. Now put that far away and you can immediately see that object is extremely small. Even the galactic center, about which you will hear, has a size, and with 4 million solar masses, has a size of only 20 microarc seconds. That's about this much on the moon. So a normal telescope would not be able to resolve that. So that must be our first goal, to build telescopes which have high enough resolution. And we know, of course, the law of diffraction. If you have a lens there, then the angular resolution of that lens is proportional to the wavelength of measurement and inversely proportional to the size. So build a big telescope and then, you have the resolution. No, that's not that simple. Because if you have looked in the dark night out there, you see the stars are flimmering. And that flimmering is called seeing. And that you have to overcome, and I'll tell you how we do this. But then actually, it's interesting that all the experiments which I mentioned to you, the optical work or optical near-infrared work, the gravitational waves, and the radio waves, all essentially work with the same technology. And that's a technology which was invented by Michelson and Mori and Young, and that's interferometry. So you take here, in this case, 4 mirrors, and you let the light combine from these 4 mirrors, then you get interference. You get these interference fringes, as we call it, and they are spaced, the narrower, the wider the mirrors are apart. So actually, Michelson in 1920 put 4 such mirrors on top of the 180-inch on Mount Wilson. You see it there, and looked at distant stars. And he was actually able, for the first time then, to do interferometry in the optical and resolve the sizes of stars. That was a sensation then, but that was Michelson. Nobody else could really repeat this for a long time. Charlie Towns was the second one, but then it took until only about 10, 15 years ago until the interferometry in the short wavelengths is possible. Okay, the radio interferometry I already mentioned, and the current leading device is called the Event Horizon Telescope, as I said, with telescopes spanning the entire globe. And the gravitational wave people, use interferometers just like that. LIGO, Virgo, Kaga. And with that, they have been able to look at masses which move around each other and then, in the end, actually produce a black hole. Now, the machine which we have actually worked with in the last years is this one here. It's Europe's large 4 times 8-meter telescope, VLT, on a mountain in Chile, in the Atacama Desert. And you see these wonderful telescopes, beautiful skies there, and we have been able to actually combine the light of these telescopes and get very high resolution out of them. Last night, we, for the first time, did the following experiment. We took from these 4 telescopes and sent 4 laser beams into space. Now, those laser beams are focused at about 100 kilometers in the upper atmosphere. There, they create what we know as a laser star, which is then reflected backwards into the telescope. And that we use to compensate the vibrations of the Earth's atmosphere. It's a very complicated technique. This technique has been in the working for about 30 years, and I have to say, I'm extremely proud about the European consortium which has been able to do this. And we get from that, indeed, images which is so good as, for sure, the 8-meter telescopes themselves, but we get even further. We can use these 4 telescopes in an interferometer to get the resolution of the size of the interferometer. So that's a 200-meter telescope. 200-meter telescope. So that's extremely good. So here is our first target, the galactic center, because as we discussed, these quasars are too far away. At least at that time, back then, it was too far. Not anymore, it turns out. But the galactic center is so close. But there's a problem. When you look up in the sky, in the southern sky in particular, you see the wonderful plane of the Milky Way at night. But where the galactic center is, it's a dirty black. You see this stuff. That stuff is interstellar material, interstellar dust. And that interstellar dust basically, weakens the short wavelength light, in particular the optical. Optical doesn't even make it to us from the galactic center. But with the technique of adaptive optics, we can actually then go to high resolution, and we see the center of our galaxy. Many, many stars there. Millions of stars. 1 million times higher density of stars than we have it around us. Also very massive stars. There's also this green thing is a radio source, which is very bright and very compact. So this is the center, we believe, of our galaxy. And so in the 1980s, first Charles Towns and his group, and I was one of the postdocs, and then later on, we and also Andrea Guess at UCLA used the technique of adaptive optics and so forth to get ever better imagery of the central region. That's interesting. That's called flying into a black hole. Yeah, time goes backwards then, it's very clear. Okay, let's go back where time was. Okay, so that's the galactic center, and there is that picture. And the idea was then, when we started this with the stars in about 1990, and the first detectors which were good enough in the infrared to measure the positions of those stars and then measure them again and again and again for years, for many years. Both groups measured for 30 years until they had a solid result of the motions of the stars. There they are. Here you can see for a few of the stars we are tracking actually the orbits of the stars surrounding Sagittarius star, that radio source. So the radio source, as perhaps suspected, is the center of the Milky Way. There's only radio emission, not much optical emission, but obviously that could be it. And indeed, the motions of the stars when they come close to the radio source are enormous, about 8,000 kilometers per second, 100 times as fast as the Earth around the Sun, or more. Here you see the data set in 1 year, 21, how we actually can see within a given year the motions of these stars around Sagittarius star, which is one of the points there in the center. And indeed, the velocity is about 8,000 kilometers per second, and that tells you something about the strength of gravity. Okay, so that is sort of the technique we're using, and then you have to have patience. And we have a very, very good team since over all these years to improve the equipment and get ever better images. I'm sorry with my fingers here, I'm doing a mess. So for this particular star here, S2, we have been able to put together a complete orbit. It takes 16 years for the star to go around. And we can see when it's close to Sagittarius A star, at a distance of about 15 light hours only, that it moves from day to day, not year to year, from day to day. And we are beginning to see the first effects of general relativity. So for instance, as you see in the bottom right there, we see that relative to Newton, where you would not expect any change in the wavelength of a line due to the mass, that when the star comes close, and that's in the center there, it's shifted to the red, so positive velocities. And general relativity is the red curve, and the data are the green symbols. So you can see we can, in a sense, quantitatively now go ahead and test not only is there a mass there, but is the mass and the effects we see of that mass consistent with general relativity to the detail. And that is really what we should do as scientists. Now, the stars move at about 1,000 times the event horizon size, so that's still fairly large. But we can also go closer. Sometimes there are little flares, probably magnetic effects on the surface of Sagittarius star. And when they occur, it's polarized emission from very hot gas, and then that gas starts to orbit at about 1/3 the speed of light. And our radio colleagues see also the distortions, the lensing of the central source. And finally, in general relativity, if you have one massive object in the center and another one orbiting it, in Newton, if there's nothing else around, that orbit would always stay the same for all times. Not in general relativity. In general relativity, the orbit starts to precess in the plane of the motions. That's up there. That's called the Schwarzschild precession. And also it precesses if there is spin of the black hole around the black hole. So there are really very well predicted effects which we You can measure this here, but down there below, you can see that compared to Newton, where we wouldn't have seen any deviations, we see what general relativity indeed expects. So the Schwarzschild precession has been seen. Now the next big step is spin. And sure enough, since last year, we have now discovered a star which comes 10 times closer than the star S2. It's only 100 Schwarzschild radii, and that allows us to probably see the spin in a few years. So all of these measurements, which I've shown you here in the galactic center, radio, near-infrared, etc., have given us a masked distribution as a function of radius, and show that that is equal to a constant point mass of about 4 million solar masses. So if you now ask the question, is that really a black hole? Then you can compare it to this diagram where a black hole is on the very left side. And all other possibilities which our theoretical friends have come up with, like conglomerates of dark matter, fermion balls, boson stars, and etc., are a little further out, not much, but a little further out. And indeed, already now we can exclude fermion balls and almost also the boson stars. In the next generation with the big telescope ELT and also the EHT, we hope to exclude boson stars for sure. And then finally, when I'm probably already under the table, gravitational waves in space with the ELISA mission will be able to see the in-spiral of a stellar black hole into a big black hole, and that will give us the accuracy to say that all of these other things are wrong. But you see, if you follow me, this takes time. Sometimes there are discoveries which you make in one afternoon. Sometimes the discovery takes 50 years, or in this case, 100 years. But I hope you follow me. We, as humanity, are curious. That is our key feature. We want to know, not to hear in social media or from President Trump. No, we want to know, and for that we have to have sometimes real patience to go with it, build ever better equipment, think ever harder, have better computers, and then find the truth. And I hope the young people here have that courage to take on this long road which you need. Now, let's go a little further out. So I've talked about the galactic center, but then in the meantime, many other objects astronomers have found in nearby galaxies that may very well be also black holes. You can't know, but it looks pretty like they are. And the interesting thing is when you plot in the vertical scale the mass of these central objects versus the mass of the center of the galaxy, the bulge mass, these are these green points. You see they're sort of a little correlated. Not super correlated, but you know, there's a scattery correlation. And that scattery correlation, everyone believes, has to do with the evolution. The black holes and the galaxies form together. And Simon White will tell you more about this. That is a feature of what we know about our universe and how our universe on very large scales formed structure. And then over time, the galaxies which formed grew in mass because they acquired ever more material from the outside. And maybe that also then led to the formation of black holes. And indeed, for the nearby galaxies, the ratio of that black hole or dark central mass to the bulge mass is about constant. This is what I said. It's likely a correlation. But here's one issue. When you calculate how fast they are growing in the local universe, then the mass doubling time scale is 400 megayears, 400 million years. Sounds like a long time, doesn't it? But actually, in the universe, it's not. Excepting at very high redshift, 13 billion years ago, there shouldn't be any black holes close to that because they wouldn't have had time. And sometimes you really experience surprises. The James Webb Telescope has all of a sudden discovered pretty big black holes already about a gigayear or even a few hundred megayears after the Big Bang. And we are puzzled. We are puzzled how that could be. Are there different ways of forming black holes? Now, of course, first off, we have to show that these objects which we have identified are black holes, like in the galactic center. That seemed like a joke, impossible 20 years ago, but it is not anymore. Because the supermassive black holes here often are surrounded by a very hot region, a so-called broadline region, which moves with a speed which is given by the size of the broadline region and the mass of the central object. And so we can actually, with the interferometer, see that rotation. And from the rotation and the size, we get a mass. And so we can measure actually the masses of distant quasars. There it goes. Here we're flying, in our spaceship at speed of light through the solar system, away outside of the Milky Way. And finally, after 12 billion years, there's the Milky Way, other galaxies surrounding. We're still traveling and traveling and traveling. And finally, there is one of these active galactic nuclei with a broadline region and a black hole. And so what we do is we take spectra with the interferometer of this rotating disk there, and when the light from that active galactic nucleus travels back all the way the way we just came, then we can, in our instrument, the interferometer, actually resolve it and see how the red and the blue parts of the spectrum are separated. Now, this is fairly superb technology, and it took us about 25 years to get there, but we are now there that we see these patterns, which are due to the rotation of a broadline region. See, that's the pattern, and then you put it onto the spectra, and there it is. And that's our inference for a distant quasar. So this is what I mentioned to you. We now are able to measure what are probably black holes at 1 to 2 gigayears after the Big Bang. So while in the local universe, these red points gave us this correlation which I showed you before, when you go very close to the Big Bang, the new measurements, both with JWST and also with our instrument, show sometimes very much bigger masses of black holes relative to the central mass. So the question is, what does that mean? Do they grow faster? And if so, why? Is there so-called super-Eddington accretion? Are there other types of star clusters which gave an advantage? Or, and that has been speculated in the last few years, very much so by the particle physicists, are there already black holes formed in the Big Bang? You see, there is almost an infinite possibility of questions. You young people, the sky is open for you to study and study the in-spirals, which I mentioned already, measure the black hole masses through gravitational waves, or to interferometry, as I mentioned to you. So in the future, we have so many fantastic machines. The James Webb Telescope now, and hopefully for some time, in about 3 years, we will have a 40-meter optical telescope in the Atacama Desert. Then this mission LISA in space, which I mentioned to you already, measuring the in-spiral of a black hole into a big black hole. This is going to be incredibly exciting, incredibly exciting. Thank you.
Thank you. Thank you, Professor Genzel, for such inspiring and really interesting presentation for our audience. Now we'll open the floor for questions. So if you have any questions, please feel free to put up your hand, and our staff members will be bringing a mic over to you. Any questions from the floor? We'll also open the question and answer session to our online audience as well. So please feel free to type out your questions at the Q&A box. Okay, we have one gentleman here at the front.
Thank you, Professor. My name is Jeff, techchannel.uk. Yesterday, I already asked you one question about the general development of the astrophysics in China and US. I want to follow up. Would you give some advice to like the Hong Kong government, the Chinese government, how the two places can improve their technological developments and in general, not only in physics, across different technologies?
Yeah. Thank you for the question. Well, okay, I'm not the sphinx of all these questions, but it's pretty obvious that astrophysics now is one of the most active and interesting parts of physics research. That was different 40 years ago when I started. My father, who was a physicist, did not think that astronomy was all that interesting. But that has changed, and the reasons are obvious. We can get at issues and problems with our technology now, which were absolutely hopeless back then. Can that go on? And there we have to be doubtful, because the machines which we have already seen now, like the James Webb Telescope, that is at the absolute limit of what the United States of America has, and I believe will be able to put into basic research. We're very, very lucky to have it. It almost died 3 or 4 times on its way, but then we have it. In Europe, we have had, I think, the one clever thing coming from the war, which is we've learned to work together and have international collaboration and not try every nation for itself to do this, because we could have never done anything. And all the great things we can do now, like these telescopes in the Atacama Desert, or also the European Space Agency, is due to collaboration. I know, and I told you yesterday, I'm absolutely full of admiration for the progress in this region and country. It's absolutely fantastic to see the vigor and interest of the young people to get themselves active in this region. That is your asset, number one. Your asset number two, of course, is if you have a government which is willing to support it, well, that's a fundamental thing. But will they be willing to support for a very long time a sustained effort which will be needed to ask some of the difficult questions? So that is something you have to build up a structure that can make that case. And then, of course, the question is, how about worldwide collaboration? We've tried this now in some cases, not always easy, because if the political and structural interests of the partners are too far apart from each other, it's very difficult to make decisions. Of course, China will and can move forward. The big fast telescope is already one example, and the space experiments are already very, very impressive. But let me just congratulate you in China and in Hong Kong for making that step, because it's really fantastic for me. I've been coming here to Honolulu, to Hong Kong for well, I've been also in Honolulu, but to Hong Kong for 20 years, and I think it's just breathtaking.
It is our honor to have you at the forum as well. So we have another question from our audience.
Hello, thank you so much for your talk. My name is Rafael Asso, and I'm a biochemist at the University of Toronto studying Parkinson's. And so I'm obviously looking at your talk from a very biochemist point of view, and I'm always thinking about biosafety and how to keep researchers safe. So, if one of us ever encountered that mini black hole as shown in your final film, sorry, I'm trying not to laugh myself, but how do you recommend, what are the safety measures in terms of interacting with black holes? Should we be interacting with black holes? And finally, who do we call if we ever find a black hole? Thank you.
Yeah. Okay, I can calm you down. The problem of an encounter with a black hole is not very likely, okay, for any given time. But let me give you an example where we humans actually have escaped deaths, at a far better way than we would have thought, and that's the Apollo mission. In retrospect, one has looked at the time occurrence of the Apollo missions and the occurrence of solar flares. And there was in particular one, I believe, of the Apollo missions where a solar flare occurred within a day of the end of the mission, I believe. Had that flare occurred then, the astronauts would have been dead. Absolutely clear and dead. So in that sense, let me say, I personally am not, and that is of course every country's and every region's decision, count on manned spaceflight. That is something which, maybe we leave it to Musk and so forth. But I think we can do unmanned probes equally good to do the difficult questions. We don't need to have people up there.
Okay, there's one more gentleman over there.
Hi there. Good morning, Professor. So thank you so much for your very interesting talk this morning. Really enjoyed it. My name is Adolphus. I'm from Singapore. I work on risk science and uncertainty quantification. And I look at your presentation and I just recall many years ago I was working on the microsat interferometer, and my mates and I, we took about days just to calibrate the system so it's precise enough to generate the interference pattern. And I was just beginning to wonder, for a large scale setup like what you just showed, and there that shot up 4 lasers and you're able to obtain some patterns with high degree of precision, and it begins the question, how long did you actually take to calibrate everything so that in this large scale it is able to produce such wonderful pattern? Thank you.
Well, the first laser star was generated in the United States as part of the military research. It was not used for very much astronomy, but then in Europe we started in the early 90s. We had the first sodium laser star going in about 93, okay? Then ESO, the European Southern Observatory, gave us the green light for the first big machine. And since then, many telescopes on the Earth have used that technology. However, what I've shown you today is indeed the first time we use it for 4 telescopes at the same time, and thereby, make such a high resolution possible for distant telescopes. And we are dreaming, and maybe that's where international collaboration could come in to make an interferometer with baselines of 1, 10, 100 kilometers. That would give us resolutions this big near the Big Bang, okay?
Any other questions from the audience? There's one more.
Good morning, Professor. I'm a researcher from Tsinghua University, and what do you think of the biggest opportunity to make discoveries about deep learning in the planetary science? Okay, thank you.
I didn't get the question, sorry.
Deep learning means artificial intelligence.
Deep
Well,
learning.
artificial intelligence and intelligent learning and so forth in computers is something astronomers have been using actually for a long time, okay? It's not so surprising for us, especially the theoretical astronomers, Simon Yu, and have been using code which comes close to this, and we are using it, of course, for our big machines. But the question often is, of course, will artificial intelligence actually allow us to do something we could not as human beings do? As a helper, yes. I'm skeptical that artificial intelligence will, so to speak, come up with something which we couldn't do. But who am I? I'm an old man. Maybe we will be surprised. Simon, do you have an answer?
Thank you, Professor Genzel, for responding to this question. Any other questions from the floor? There's one more gentleman just put up his hand.
Good morning, Professor Genzel. Thank you very much for your interesting presentation. I'm Adrian. I am an astrophysicist at the University of Cambridge, working on analyzing gravitational waves emitted by black holes. So I consider myself half a black hole physicist, but one difficulties that I encounter is how to explain to the general public why we should care about black holes. So would you have any advice about when I try to popularize or explain my work to the general public, how I should proceed? Of course, I guess you won't just show the public the last movie that you have just shown, right? Thank you.
Thank you for the question. Okay, well, I'm not almighty in my knowledge, but it is certainly the case that I'm giving many, many talks per year, about 40, 50 talks, and 30 to schools, and I can tell you, the younger the better. Yeah, I mean, the young people, 10 to 15, even 7 to 15, you know, they love it to hear all of this. They may not understand everything, but they love to hear it. And in this curiosity, that's what makes, as I said, that's what makes us human. And you have to be curious because you have to do something difficult. And difficult means you have to put your mind and perhaps your fingers and whatever else to it for a long time. From nothing comes nothing, okay? No lunch out of, you know, thin air.
Yes, we have one laureate right there that would like to share a few words. May we have? Thank you.
Well, from your challenge, a couple of thoughts spring to mind about artificial intelligence and what we can learn for it. Because as we all know, everyone is thinking about this and all the things that we see, magic that seems to come from ChatGPT and large language models. And the first thing which springs to my mind is large language models summarize all the data which they can put together and analyze. And so they're summarizing for us in some way the sum of human knowledge as written. And that includes the correct results and also all the rubbish. And what you get back is a summary of everything, and it takes considerable judgment to decide what is rubbish and what is there. So there's an enormous danger in taking what you get back from the language directly. The second thing is, which I think is a deeper thing, is that we're talking about new knowledge. And we don't really understand where scientific insight comes from or any other kind of human insight, but it seems to be a capability of the human species to generate what we at least perceive as new knowledge. And I don't think you can get that from a large language model. So maybe the most important part of scientific research can never come from this kind of algorithm.
Thank you, Simon. I think that's very helpful. We had every year there's the conference of the Nobel laureates in Lindau in southern Germany. And 2 years ago, we had 35 laureates on stage. And the question was, do you believe that artificial intelligence will give us a major new insight and discovery? And I would say out of the 35, about 10 said yes. The rest have said no. Your judgment. I mean, it's a guess. There are extreme dangers, you all know, politically. And there's the extreme difficulty for young people. How do we old ones, if you want to judge your work, do this if you give us an application written by ChatGPT? silence. Okay, thank you.
Thank you so much. We have time for 2 more questions. So let's see if anyone would like to exchange your ideas with Professor Genzel. There is one participant.
Thank you for a nice talk. I was wondering, at the beginning of your work, how did you, make sure this is the galactic center? How pinpoint the galactic center?
Yeah, no, I think that's a very fair question. In fact, I should tell you that there is no point that is the galactic center, because the black hole mass, which 4.3 million solar masses, is embedded in the galaxy. So in a way, it sits in a position which is part of the gravitational interaction with the entire galaxy. So we can compute that on average, that 4 million solar mass will, move around stochastically at about 2 centimeters per second. Not that we can easily measure this, but no. But this is not the question the answer to your question. I mean, the idea that it might be the radio source came on very, very early on. Okay. And was confirmed by a number of the properties of the radio source. Also, in comparison to other active galactic nuclei, where there's also the radio emission is sort of a signal, so also some X-ray emission. So this is sort of a signal that this might be the region you might want to look for. But then, as we were beginning to measure gravity, then it became clear.
All right, any other questions from the floor? Yes, there is one young man over there.
Hi, this is Anthony Chan, specializes in computational imaging and super resolutions. I also attend a separate tenure track lecture by Professor Katie Bowman in Caltech, who's also happened to be a major player in the Event Horizon Telescope. A question is more about the hidden figures behind all the international collaboration for investigating black hole and visualizing the
Sorry, I did not get what is it?
My question is about hidden figures and visibility as an engineer and in a PhD. So not a lot I heard about the stories about the Western digital hard drives, 25 petabytes flown daily from the telescope site to the Max Planck Institute, and they are helium filled. With all this technological breakthrough and scientific challenges in order to support a single figure in the astrophysics journals. As a PhD in engineering, what's your advice to them to gain more visibility and to write about their own stories in the international collaboration, especially in the Event Horizon Telescope scale program? Thank you.
I'm sorry, I failed the test. I don't understand what your question is.
I think the keywords from his questions were hidden figures and visibility. Am I correct?
Pretty much, yes. That's excuse my strong accent here. Thank you.
The visibility of
as an engineer, PhDs. Right. The hard drives were helium filled and fly daily from the Event Horizon Telescope to the Max Planck Institute for follow-up data analysis. And these are all the engineering charges that deserve a team of PhDs there. But they don't write papers, right? Thank you.
Yeah, well, okay. I mean, I think in our experiment, we, of course, have also a large team overall, maybe if you count ESO and the other countries, maybe 150 people. But we are an integrated team, so the engineers are on the scientific publications as well as the scientists. We don't make any difference there. Okay. Now that is also disputed by some of our colleagues because it makes these huge lists of publications. But I have grown up under the tutelage of Charles Towns to have the greatest respect for engineers and what they do. Okay, without them, we couldn't build what we build. Okay, and so in that sense, the visibility has to also be given to them, and the triumph has to be given to them. I showed you this measurement with the 4 lasers 2 nights ago. And yesterday at noontime, Hong Kong time, my computer started exploding as the entire team and ESO started jubilating and congratulating each other. Now the Event Horizon team is a more difficult thing. I cannot understand it in detail. It has some features which I'm a little uncertain about how they are doing it and what they are doing. They're not a oneness as I think you might try to be.
Thank you. And one last question from the audience, please. Yes, there's one.
Hello, my name is Zishan. I'm a PhD student studying galaxy evolution. So my question is also about how to communicate our field to the general public. I think you made a great comment about when you talk to the young students, like they are very curious. But I'm trying to ask him from the other angle. What about those older people? They probably in the position of power. They are kind of like the funding source to our research. They could be government officials. They could be private donors such as real estate developer. So how do we convince them our research about black holes are important to helping the countries to develop?
Well, okay, look, I have as a European, as a German, the advantage that I don't have to fight for donors as I would have to do in Berkeley. But my wife is sitting there. She's a pediatrician, and so she has managed several times to hook me up with her PhDs and her doctors. And I thought they were very interested too, weren't they? They were. I don't think it's just a question of how you do it. I often use music, actually, as a sort of continuation. And of course, if you come and explain dry formulas, that's hard on everyone. But if you make it fun, I mean, then people like it. Yeah? And the kids have the advantage that they still do this.
Maybe you can create a very interesting presentation to convince your future donor when he's very young. All right, thank you very much, Professor Genzel. Let's put your hands together to thank Professor Genzel for all the inspiring presentation and all the insights that she has provided. Thank you. We're truly honored to have Professor Genzel with us at the forum and be inspired by his presentations. And I'm sure we all look forward to further interact and exchange more ideas with Professor Genzel in the coming days. So now, ladies and gentlemen, we'll have a 20-minute coffee break. Refreshments are available at the back of the hall. Please be returned at the hall before 11 a.m. for our panel discussion later on. And we'll see you later. Thank you. Ladies and gentlemen, our panel discussion will soon begin. Please return to your seats. Thank you. Ladies and gentlemen, the following session will soon begin. We'd like to invite you to return to your seats and be ready for the upcoming panel discussion. Thank you very much. Ladies and gentlemen, the upcoming session will begin soon. Please return to your seats as quickly as possible. Thank you. Hello everyone, welcome back, and I hope you're ready for the upcoming session. In the coming hour, our distinguished scientists will talk about biological intelligence, how life sciences inspire new frontiers in data, diagnostics, and discovery. We're excited to have 2 laureates joining us to share in our panel discussion, and the session will be co-hosted by our very own local scientist, Professor Danny Chan, followed by a Q&A session. Professor Kazutoshi Mori and Professor Peter Walter were awarded Shaw Prize in the discipline of Life Science and Medicine in 2014, in recognition of their outstanding contribution in the discovery of the unfolded protein response of the endoplasmic reticulum, a cell signaling pathway that controls organelle homeostasis and quality of protein export in eukaryotic cells. Now, may we invite our speakers and moderator to this stage. Let's welcome them. We have Professor Kazutoshi Mori, Distinguished Professor, Kyoto University Institute for Advanced Study. Let's put your hands together to welcome him to the stage. Next, we have Professor Peter Walter, Professor, Biochemistry and Biophysics, University of California, San Francisco. And hosting the session for us, we have Professor Danny Chan, Emeritus Professor, School of Biomedical Sciences and Director of Development and Education of Research Integrity, the University of Hong Kong. Welcome again. Please take your seats. Now we're ready to be inspired. Over to you, Professor Chan.
Okay. Hello, good morning, and welcome to this session, plenary discussion. Today we have 2 very distinguished laureates with us, Professor Peter Walter and also Professor Kazu Mori, who will discuss with us on the topic of biological intelligence and the role of and the role of how life science inspire new foundation in data, diagnostic, and discovery. From the perspective of the 2 laureates. And I'm also super excited and honored to have the opportunity to chair this discussion session. Now, biological intelligence is a huge topic, which will probably take hours to discuss. But maybe perhaps we should first touch base on the later discussion. And we will first focus a little bit on how life science inspire new frontier in data, diagnostic, and discovery. Perhaps from the 2 laureates, Peter and Kazu, if I can address you by your first name, to keep this forum a little bit informal, as we all already dress quite formally as basic scientists. Yeah. Now, yesterday, I think Peter and Kazu gave 2 really wonderful lectures on the mechanistic insights and perhaps potential translation opportunities from their favorite topic of the unfolded protein response, or the UPR, from which they have both won and share many prizes and counting. And of course, the Shaw Prize. Now, on how life science inspire new frontier, Peter and Kazu, I am sure the audience would love to hear a little bit from you, a bit from the historical background leading to your discoveries. So my first question to both of you is what made you interested in cell biology in the first instance, And what make you, the decision making in choosing this topic to focus on the unfolded protein response, yeah? And perhaps maybe a little bit on your discovery process, whether it is a targeted discovery or whether it's something serendipity. Yeah? So maybe perhaps I begin with you, Peter. You could share a bit of your early career research and leading to that discovery.
Yeah, so I grew up in Berlin, in Germany. And then I started studying chemistry. I went to the United States at Vanderbilt University, get a master's in organic chemistry. And from there I went to Rockefeller University, where I got my PhD with Günter Blobel in serology. And what impressed me when I first visited as a totally naive chemist, entering the laboratory of serology, there were these beautiful pictures, electron micrographs of George Pallade on the wall, of how beautiful and how complicated cells are. And I think that sort of motivated my transition from chemistry, which is pretty much more an engineering discipline. We make molecules, we synthesize them, to biology, where we are just faced with the products of evolution, and we are trying to make sense out of them. So it is a path of continuous detective work. And since evolution takes pieces that are lying around and puts them together in different ways to make them useful for a new function, these are Rube Goldberg machines, where the rules are not really deterministic of what is being used for what. So that the spectrum of discoveries you can make is absolutely enormous. So I worked in Günter's lab on protein translocation into the endoplasmic reticulum. I discovered one of the components based on my graduate work, that is the signal recognition particle, and characterized its function, basically biochemical. And with that accomplishment, I got offered a faculty position at UCSF, where I then started my own lab and continued my career for 40 years. And in this course of transitioning to a new place, I was exposed to a new environment, new mentors, senior faculty members, and one of them was Ira Herskowitz, and Ira was a yeast geneticist. I took his genetics course as a young assistant professor. I took a class with him and learned genetics. And now my lab was ready and was eager and wanted to do some genetic experiment. So the unfolded protein response came out of the desire of two graduate students in my lab who wanted to do something genetic in the lab. And the question we came up with is that proteins, as they enter the endoplasmic reticulum, which we were the experts at the time, now they have to fold up. So how do they know how does a cell know that it has enough folding machinery to do so? There was previous work, some from Kazu's work, that the transcription machinery in the nucleus provides the proper amount of chaperones. So it makes more mRNA for chaperones. They enter the endoplasmic reticulum. So somehow the endoplasmic reticulum had to tell the nuclear compartment what it needed. There needed to be a feedback loop that establishes the appropriate amount of folding machinery for the amount of proteins that have to be folded. So this homeostatic event cannot happen just randomly, so there must be communication, and we set out a genetic screen to identify components on that communication pathway. And that screen led to IRE1, which then turned out to be one of the most marvelous and beautiful enzymes. We are still working on it today. There's going to be a poster by San Wen Hu in the lab, my lab currently, who will present this tomorrow. It's one surprise after the other, and we keep working on it. And it's a single transduction device that sits in the membrane of the endoplasmic reticulum, figures out with one end what's going on in there. Do we have unfolded proteins? Do we need more chaperones? And then it transmits that information across the membrane into the cytoplasmic space, from where it then activates the transcription machinery that provides the appropriate gene products to make more of the chaperones, to take care of the proteins. So it's a very beautiful little loop of signaling, and it uses very unusual machinery and bits and pieces that have kept us busy for literally decades and have produced many PhDs.
Thank you, Peter. Ka-shing, perhaps you can share your perspective?
Nearly 50 years ago, I was a freshman of Kyoto University. At that time, I did not like biology. Textbook seemed very too descriptive. So I was a student at the Department of Chemistry, a faculty of engineering. But when I was reading newspaper, I found an interesting term, molecular biology. What is it? I searched many articles by myself and found that idea of central dogma. DNA carries genetic information, it's transcribed to RNA and translated to protein. And this genetic code is conserved from E. coli to human. I was amazed that biology has a beautiful, simple theory. So I wanted to learn molecular biology. So I changed my major from chemistry to biology, just like Peter. But molecular biology is the most advanced science. So not so many laboratories can work on it in Japan. So I chose biochemistry. I was biochemistry nearly 10 years and got a position of assistant professor in a local university in Japan. I kept working on biochemistry, but I could not stop my passion to the molecular biology. So at age 30, I decided to go to the United States to learn molecular biology. So my boss at University of Texas Southwestern Medical Center, Joe Sambrook and Mary J. Gasing, accepted me as a postdoc in 1989. 1 year before, in 1988, they published a paper in Nature that showing that our eukaryotic cells are equipped with an interesting system called UPR. So that's my start. So when I become a postdoc in their laboratory, UPR system exists, but no molecule identified at all. So I was quite fortunate. I could start from beginning. Then it's a transcriptional induction system. I analyzed promoter region of chaperones and discovered cis-acting element responsible for the transcriptional induction we named UPRE, unfolded protein response element. Then published a paper in EMBO Journal, 1992. Then usually, next step is purification protein capable of binding to UPRE element. But I was a biochemist. Purification protein means hard work in a very cold room for a longer period. I didn't repeat biochemistry again, so I wanted to challenge new things in United States. So I challenged yeast genetics. I isolated a mutant which unable to induce chaperone even in the, under the condition of ER stress. And I screened 100,000 colonies and worked for half a year. I got 3 mutant cells. Then it took another half year to get gene that defective in that mutant. Then I got IR1, or Peter said, we had that same molecule. I had IR1 as an ER stress sensor. Then it's published in 1993. Since still I'm working on this UPL system, I switched from E-cell to mammalian system, and now we're working on the mammalian UPL, or using fish model, still working on the UPL system, biology and physiology of the UPL.
Thank you, Peter and Ka-shing. So having discovered the very first sense of the UPL obviously is very important and has opened up a huge area of research for yourself, I guess not only for yourself, but also for many other researchers like myself as well too. So I wonder if you could share with us, how you take your initial discovery and to further, to take on that challenge to look for more information and how you would inspire from that. Maybe I'll start with you again first, Peter.
Sure. So the first screen was done by 2 graduate students in the lab, Caroline Shambu and Jeff Cox. They collaborated wonderfully for the screen. And once we had the first component, then the discussions began. Now, what shall I do and what shall I do? How do we divide our work so that the absolute connectivity of doing a screen together didn't last any longer. So we had to somehow divide the project to give them individual freedom to develop their own path forward. So Caroline chose to be more involved in characterizing the molecule we had already identified. And Jeff said, well, I'll just do another screen and find the next component downstream. Because once we have this signal transduction device sitting in the membrane, it has to talk to something that then talks to the transcription factor. So we did another screen and we pulled out the transcription factor that happens to be activated by IA1. It's activated in the most fascinating way by having a very unusual splicing reaction. So the field started exploding we exploded in different directions. We stayed in yeast for the longest time and determined the mechanism, which then beautifully, most of the salient features held true in the higher metazoan cells and in mammalian cells and in fish. So it became it spread out into many avenues that became complementary to one another.
So after publication of IRE paper in Cell in 1993, I went back to Japan and I got position as a researcher in an institute which existed only for 7 years contract. Then next target is the transcription factor. Then, again, I did not want to purify a protein, so I managed to try to identify factor by molecular biological techniques, but it's difficult. I struggled for a while, but one day I came up with the idea of one hybrid screening. And then this idea, one hybrid screening is a modified version of 2 hybrid screening, which works only for transcription factor. Then this screening works well, and I got HAC1, like Peter had Simon. Then find the splicing of HAC1 mRNA. But one hybrid, as I mentioned, one hybrid screening is a modified version of 2 hybrid screening that can be applied to any other species. Then using the same strategy, we could get mammalian transcription factor after identification of 6 acting elements in a mammalian chaperone promoter, which explain simultaneous induction of many chaperones. So now then that identified 86 BPN, 2 key players in the mammalian UPL.
Ka-shing, perhaps I'll ask you a further question. Because having spent time in the US, you decided to move back to Kyoto, right? And you think that it was the right decision. It was a very important decision to move back to Kyoto. So you had some premonition that U.S. going to get into trouble, or is there a particular reason why you wanted to move back to Japan to carry on your career?
So when I was working on the IRE at the last stages, my boss Joe Sandberg asked me, "What do you want to do after publication of this paper? If you want to stay in the United States, I can give you time and place for job interviews. But if I keep staying on UT Southwestern, it's easy, and I will happy to do that. But to get the position, I have to go somewhere else, an unknown place, and create a new relationship with new faculty members. I thought it's kind of it was difficult. Then I decided to go back to Japan. But I found a position in the institute. It's well funded. The new boss, Dr. Yura, is a gentleman. But when I want to isolate transcription factor by multi-copy suppressor screening, but he said no. Only that time he said critical, but no, because IRE is a kinase. So if we use multi-copy suppressor screening, there might be more kinase. You may get kinase, but never reach transcription factor. You have to find a method by which you can directly get transcription factor. It's a difficult job, but I managed to came up with the idea with one hybrid screening. Then that kept me going. So that was very good advice.
Thank you. So Peter and Ka-shing, since that discovery, you both have published extremely well and continue to discover the mechanistic insights into the process. And you two have become really good friends as well, right? Research these days, research is supposed to be fun, right? Full of curiosity. But many young investigators these days actually feel research to be highly competitive and a little bit stressed out as well. So was this also an issue for you? Or it was, you consider it as a synergistic discovery path?
Well, shall I start?
Yes, please, Peter.
I think it was fun competition. So
Peter was a big war to meet.
We were definitely competing with each other, and I think it was absolutely productive and fantastic in the end, because we were all out. We were making many serendipitous discoveries here that were not the usual mechanism that happens. We have a very unusual splicing event in mRNA for transcription factor that IR1 is initiating, and so on and so forth. So pretty much everything we found, or what Kazu found, we wanted to prove wrong in the respective labs. And then we corrected each other at some point in the past, and we confirmed each other in other ways. And by having this sort of dichotomy between the two different groups, trying to do the same thing, but never publishing a paper together, everything was, even no matter how strange the mechanism seemed at the time, it was accepted by the field very quickly as being, yes, this is the way it is. So science moved much faster, much more directional. We didn't bark up the wrong trees for too long. And I think it turned out to be absolutely a fantastic relationship, even so there were a few painful moments, but it was overall great. So don't shy away from competition. Just make it friendly, right? It's not personal. There's nothing personal in it. You don't start hating people. You just go for what their science contributes, and you argue on with data.
Should you like that?
in 1990s, Peter was a big wall. So I have to keep working on hard. And when journal Cell come to our institute, I checked all index. Oh, I don't see Peter's paper. Okay, then I read papers carefully. But just Peter said that this competition derived this field dramatically forward. And then I could move on to the mammalian system because ECUPL is too competitive for me. I found a way to go by myself.
Right, so the message to the young people is that networking is very important, collaboration is very important. Don't keep information to yourself, share it. I think this is the way to sort of, work together synergistically, just like what Peter and Kazu do. Right. Okay, now it is actually well recognized that there are many diseases associated with the protein folding and cellular stress pathways, right? So given, I guess a key question is that how your discovery in cell biology have influenced understanding of disease and innovation in diagnostics and potential therapeutic outcomes. And maybe any clinical breakthrough that you may like to share with us, from your own perspective or even from your own research. Maybe Peter first, and then Kazu, if you want to add to that.
Yes, so we started out, me being a chemist, slithering into cell biology, we started out with very basic foundational research. Trying to figure out mechanistic details. And it eventually became clear as the field emerged from our work, from many other works, that protein folding and the whole idea of quality control in the cell is tremendously important for many disease settings. And then mostly through collaborative work, we started engaging with groups to apply our knowledge to see what we can do about these different diseases. Cancer comes to mind. Cancer cells evolve in your body in a certain spot. They become ever more genomically unstable, so they make many mistakes in the proteins they produce. That turns on these surveillance pathways. Not just the unfolded protein response, but also heat shock response and others. And so the general image emerged here, right? That as we fix these stress pathways, as we modulate them, learn more ways where we can potentially drug them, we could do good things. Because in cancer cells now, these pathways are activated. They make decisions, life-death decisions for the cell. Most of us don't have cancer because the cells that have these abnormalities are programmed to die. And if these events go wrong, because the unfolded protein response does not make that decision correctly, then we get cancer. So the cancer cell then uses these pathways as a band-aid to cover up, to have enough chaperones, to cover up all these passenger mutation, proteins that have these passenger mutations that are otherwise seen as unfolded proteins and as detrimental. So if we take this away from them, we can selectively kill cancer cells. So we engaged by now, it's a over 10-year long collaboration with Avi Ashkenazi at Genentech, who is developing new ways of chemotherapy based on blocking the unfolded protein response, and is now in preclinical development and looks very promising. It's very non-toxic because these stress pathways are only activated in cells that are unhappy, that have stress, that have misfolded proteins. So we can select these drugs and these inhibitions pretty targeted to the diseased cell and eliminate it from the body. So that works great. And then as the field of protein quality control emerged, the other branches of the unfolded protein response emerged, the PERC branch and the ATF6 branch. We found very similar opportunities in these branches. So blocking the integrated stress response branch that is mediated by PERC, among others, we got into neurodegenerative disease, and that's what I talked about yesterday, showing a little small molecule that we isolated that shows a great promise of fixing various cognitive defects in a numerous set of diseases. So again, we are approaching it disease agnostically at the beginning, trying to figure out what are the mechanisms by which things work. That gives us handles of surgically interfering with these pathways. And then through collaborative efforts of people who are having these disease models in their labs, we can advance it into potential therapeutics in the future. And again, this small molecule that we discovered in the lab is now in clinical trials for a variety of cognitive disorders.
Yes, medical doctors are quite busy by taking care of many patients. So medical doctors do not touch area field with mechanisms unknown. But our basic researcher investigated a system and clarify the mechanism. So by doing that, we can show marker for ER stress, splicing of XBP on mRNA, or phosphorylation eukaryotic translation factor, or just show the marker. Then medical doctor checked this marker is up no in disease they are looking for. So then many papers come out showing relationship with disease and ER stress. So basic research is really important to open up new diagnosis, treatment of diseases.
Thank you. I think Peter, yesterday's talk on ESRAP was fascinating. You know, this actually makes me think about what I should take some of these ESRAPs to improve my cognitive function. It's super amazing. So we know that a cell actually needs to make many decisions, right? And none more so than to live or to die. And the UPR, in a way, is actually central to such a decision. So given the topic of the panel discussions is on biological intelligence, I was wondering whether perhaps I can get your view on your thoughts on biological intelligence, and can a cellular decision making can be considered to be intelligent per se, or is this to be at a much higher level of an organism? So can I get your thoughts on this? Maybe again, Peter, start with you, and then Kazu, if you have anything to add.
Yeah, I was sort of surprised by the term the intelligent cell being used. So as I said, I trained in chemistry, so for me, a cell is a collection of molecules that all obey the rules of thermodynamics, and as such, their behavior is deterministic depending on the concentration of things we have and who is seeing each other in the right place at the right time. So I would say it's really in the language here. What do we consider intelligent? So in my view, a cell is not intelligent. A cell makes appropriate decisions depending on what the programs are, the molecular programs that it carries out. And these decisions have stochastic elements in them. So the cell death is always a fraction of what's happening because of the molecules that are available and that are doing these things. So the decision in the end is pretty deterministic in the population, and and this is evolved in a way that the ribosome is just good enough to make proteins with high enough speed and high enough fidelity. It doesn't need to use intelligence, right, to decide whether it should abort protein synthesis or not. That just happens because the way the molecular components interact with one another. So I think the term is overused, and we should leave real intelligence, which for me involves making a creative solution, right? That is, and I haven't seen it. I haven't seen a cancer cell making a creative solution to become better.
So it's more of an existential decision rather than...
Yeah.
Ka-shing, do you have anything to add to that?
I asked my colleague, please ask AI how cell decide survival, death under ER stress condition. Answer is depends on duration and strength of ER stress. If duration is become longer and strength become heavy, cell decide to die. But it's like no decision, like just heavy ER stress overwhelming cell, and they have to die. But I don't. There may be a switch, real switch from survival to death. So if there is a switch, that might be intelligent. So we are looking for such a switch. No mechanism is not always activate from the beginning. Not activation in the data time or cell decision time. So still we need to find a switch, I think.
Okay, so I do agree that intelligence is kind of overused, in particular artificial intelligence. We touched a bit on this in this morning's discussions as well too. But certainly, AI is having a huge impact in our daily life, and the same in scientific research as well too. Because many of my colleagues now actually are using many tools they call AI. None so in protein biochemistry in terms of protein folding, for example, AlphaFold, AlphaFold 3 and so forth. But that is really leveraged on, decades and decades of really hard work, right? Looking at the structures and so forth before we actually come to that. So really in terms of, it's leveraging on information rather than artificial intelligence per se. It's using information that we have to produce information. Now, a dream of cell biology like myself is really to develop an artificial cell. The dream is to try to understand every component in the cell, every molecular interaction, every assembly, and so forth, and such that we have a digital cell, and if we can perturb certain things within that and see, with a specific perturbation, we'll now be able to understand what the outcome would be. My question to you is that, do we have such information? Is it possible? Do we have enough information to arrive at such artificial or digital cell? If not, what more do we need to get? Again, Peter, maybe begin with you.
Well, I think we're getting there in terms of information that we have now opportunity with new methodology to get huge amount of information, more than we can handle with a human brain. Wolfgang's talk yesterday, when you look at the tomograms, they basically contain the information where every cell, where every component in the cell is. We just cannot distinguish them yet, unless they're big and like the proteasomes or the ribosomes stand out, and we know what we are looking at. So we will get there eventually. The problem with the data out there, on which AI relies, is of course that, and I think was said this morning and was said yesterday by Professor Mack, that probably 95% of the data out there are crap. And AI will not be able to distinguish what are the crappy data from the good data, right? And there are many, there's this gray zone in between, that things being done in different cell types may lead to opposite results because of some very trivial explanation what makes these cells different from one another. And all of that needs to be integrated at some point. So I think we are far from being able to build a virtual cell. We will be able to build virtual models that allow us to have a coarse-grained understanding. If we tickle this receptor, this will happen in terms of transcriptional program. And if we have this transcriptional program, this will happen. But it will take a long time to really be able to then zoom down and say, these are the molecular events that happen. These are all the components that are involved in that. Because one little thing, right, one little phosphatase that takes a phosphate off that we don't know about can change the picture entirely. And so we need to do both, right? We need to keep on creating data. We need to make sure that the data we produce is accurately described, so what the weaknesses are, what the strengths are, what the cell types are, what the time points are, and so on and so forth. All the conditions, all the stuff that's now shoved into supplementary material and becomes so important for the condition will become more and more important because now AI will actually read that stuff and bring it back and allow us to integrate it into our models. So it's a tremendous tool of doing all the reading for us and the integration. But we have, whatever comes out of it, we have to see with a grain of salt and put it to the experimental test.
Do you have time to add to that?
Yeah. AI really helps researchers. AlphaFold can predict three-dimensional structure, but now new AlphaFold can show protein-protein interaction. It very helpful for our story moving forward. But I have two concerns on the AI direction. Because one is, Peter said, there are many bad papers. And how AI can discriminate good paper from bad papers. So if you rely on the bad paper, the direction going wrong. And second is experimental condition is so diverse in biology. Many trial and error is required to find a good way to go. But AI can do, and then during such trial and error, there may be jump or a good idea. So AI can do or not, I'm a little skeptical.
Thank you. So obviously, I think we probably must use AI as a tool, but don't over-rely on AI. We must understand what is true and what is rubbish, right? So we still need to think that from that perspective. I know time is up. I would like to close up with one last question. We now know that a cell can get stressed out and can sense it and make existential decisions. Unfortunately, I think the changing in research culture and now politics and the competitive nature of research also places a huge pressure on many, if not all, the junior researchers and scientists. And they are all stressed out quite a lot, okay? So I wonder if you could provide some advice for the budding young researcher out there, how to actually best approach this ever-changing research environment and demand from our senior management and regulators. Final comments, please.
Well, think what the cell would do, right? It will turn on the stress pathways, and the stress pathways will try to bring things back into balance or kill the cell. You don't want to kill yourself. So you only have one option. Bring your life into balance, right? And see things in perspective. I had graduate students who were working incredibly hard and who were going nowhere with their projects because they didn't take the time to think. They didn't take the time to think of what was the important next thing to do. They just kept on doing, like hamsters running in a wheel, going nowhere. Don't put yourself in that position ever. The other thing is you need to realize that the profession we are in, 95% of what we do fails. And that is good, because if more things succeed, then you're only doing the obvious. So you want to challenge yourself and be at that level of risk-taking. So make sure that your life provides a balance to that, a hobby, do some sports, and just bring your life into harmony, and the stress will go away.
My advice is, I talked yesterday, find your own strategy to make progress. It's really important. Getting a good strategy change your life. So I hope you can find your own way to go.
Well, thank you. Thank you, Peter and Kazu, for this advice. I guess, I think a balanced work-lifestyle is very important. 7/24 in the lab doesn't necessarily bring you what you want. Time to think is more important, right? I think that the classic thing is think before you do, right? Now I think
but sometimes you need to be there at 7:20.
Yes, yes, indeed.
Just use it selectively.
Yes, indeed. I think what I'd like to do now is actually open a Q&A session to the floor. I'm sure there's lots of eager junior scientists out there who would like to ask questions to both Peter and Kazu, so please. I think the lady with the mask got her first hand up, so please.
Thank you very much for the wonderful panel discussion. I'm Rafaela, so once again, and I have something to follow up regarding the limitations of AI, especially in terms of studying protein biochemistry. Just because from my own research on Parkinson's disease, there's so much stochasticity and just with, as you said earlier, like random cellular environments influencing protein misfolding. And in my own research, actually, even if you keep everything the same, just random proteins doing random things themselves could cause completely different results. And so my question to you is, because experimental science will continue to be so important in the context that AI cannot fully predict these things, what are some future kind of techniques in terms of bench science for protein biochemistry that you predict will be very useful for expanding the field of trying to understand protein misfolding, protein unfolded response, stuff like that? Thank you.
Yeah, I think a good angle in which to think here is that most data in the structural protein database are still derived from crystallography. Right? We are now going into a predictive mode from that through AlphaFold. But we are also having new techniques with single particle cryo-EM, which gives a more native state of protein in solution. And what we are learning there is that proteins are not locked into a certain conformation. If you do molecular dynamics on a kinase, it looks alive, right? The surface is fluid, it constantly wiggles, and so every protein we have is an ensemble of states, of many, many different states that are interconverting. Some of them are good, some of them are bad, some of them just happen to be there. So by gaining more and more appreciation for that fact, I think a lot will come out of applying molecular dynamics and exploring this randomness, right? When do you fall into a trough? When are you stuck in one reaction or another? And what influences that? There's the stochastic element, and then there's the butterfly effect, right? That in climate prediction, that a tiny little change here can have a huge change here if you just change the trajectory. So as we're gaining more understand and appreciation for the fact that things are kinetically controlled and to a large degree statistic with many, many, many decisions that are constantly being made, or different paths being chosen. We're going to get a better appreciation for what we are up against.
Okay. Another question? Could I ask you to actually keep the question short and concise? Yeah? Please, lady at the back, got her hand up.
Yes, my question is, did you have any apprehensions when you were about to switch from chemistry to biology? And would you recommend for young scientists to expand their research area during their early careers?
Young scientists probably needs good mentor first. To find an interesting field. If you are interested in biology, then you don't hesitate to change major to something from something to biology. Then find a good mentor. Then a good training, and then determine what to do after getting PhD. It's most important thing after PhD is find a direction, which field you wanna go. So that's stepwise fun. Find a good major, and find a good mentor, and good education, and find a way to go after getting PhD. So my advice is that answer to your question.
I think it's one of the privileges we have in our careers, right? That we can reinvent ourselves, and that we can constantly learn into new things, right? Yes, I had apprehensions. At the beginning, you feel like a fool because you don't understand anything, right? You have to learn the lingo, you have to, I mean, you're in a totally new field, and this is, it's risk-taking, but it's also tremendously rewarding as your appreciation of the complexity of biology increases.
Thank you. We have quite a few questions online, and I'd like to actually pick one of them for you guys to tackle. How do you think the emphasis on translational research affects basic science? Is there a risk of losing curiosity-driven exploration?
In biology, curiosity-based research leads to some clinical success in the many years later. But so find basic research is important for new development.
Peter?
I mean, the basic research provides the foundation for the translational arm of clinical medicine. CRISPR came out of bacterial immune system, right? Telomerase came out of slime mold, the discoveries. So this is something that our funding agencies need to justify what they've done with the money. They need to provide more, bring in more money by in the United States, providing Congress with nice examples of how health has been improved. So I think basic science, basic discovery, foundational research is constantly endangered because it's seen as here's a scientist playing around and not knowing where they go, and not really mission-oriented. So it is a constant struggle to provide the examples of where has basic research been the important piece in a translational in a translational accomplishment. So that's why we're learning all this lingo. When you write a grant, right, you need to write down where this is going to go, which is bullshit. Because for all the things that we have done, we had no idea where this would go, and this only came when we had the data. So somehow, I'm not sure how we get out of this devil's cycle that we're currently in. So yes, there is a danger.
Yes, thank you. I guess there needs to be a balance. I think, innovation in technology and focus on translation is important so that we can translate what we discover in the lab. So we have senior management here, and why I think we should push for translational innovation. Let's not forget curiosity basic research that has driven many of the important discoveries in the past. I'm sure it will be so for the futures as well too. So back to, yes, the lady's had her hands up for quite a few times, so please, the lady at the back.
So hello, both professors. My name is Irina, and I work on dementia prevention as a therapist. So Professor Mori, you mentioned that you find your strategy to make progress. So can you share a little bit more about what your strategy is? And throughout the process, is this strategy developed before you start, or it's more like in trial and error? Thank you.
I came up with the idea of one hybrid screening to identify transition factor I'm looking for, but nothing produced anything. So I thought I came up with the idea by myself. I thought I'm a genius, but that in the United States, we had no journal club in the John Samuel Club. So we didn't have enough time to read a paper. So Japanese scientists, get together once a month and show the new paper, new interesting paper. And then one guy showed one hybrid screen in that discussion. At the time, I didn't know how important it is, but some input in my brain. Then after coming back to Japan and struggling for why to find a way to identify factor, then that idea come down to me. So you have to read many papers. It's better to read many papers to get idea. So you cannot produce nothing from everything. I think it's by reading many papers, you have to train your brain. It's my advice.
And talk to many people. That's often faster and more fun, certainly, when you do it in the bar.
I understand the time. Maybe perhaps we have one last question online that may be relevant on the issue of intelligence. So the question is, what role do you see AI playing in hypothesis generation, not just data analysis?
I think it's a powerful tool. The AI will not generate the hypothesis, but you will generate the hypothesis in an AI-assisted way. When AI digs through large data sets, right, and provides associations that we were not aware of before, we can build them into hypotheses that we then can test. Right? There's enormous data now that there is huge data banks becoming available for human mutations. Right? I mean, we all carry so many mutations. And the statistical links that are being made to phenotypes are becoming stronger, and sometimes you find that there are associations with something that may be worthwhile testing. So you build a hypothesis on that. You ask the programs to dig through these databases and see whether something correlates with your phenotype of interest. And then you have to do the experiments and see if it works. So yes, I think it can help tremendously when used cautiously. You don't ask it to put out a hypothesis. You just ask more subtle questions that then lead you to propose what is in your range of interest.
Okay. But I guess at the end of the day, those you still really look at those how those hypotheses are derived from the data that are out there, right? So even though the AI has put forward a hypothesis for you, you also need to really check and double check yourself that the data is accurate, right? Okay.
Absolutely.
Sorry, go ahead, Peter.
I was just agreeing with you.
Okay, right. So I think time's up. Unfortunately, I know that many of you still got many questions. I have to bring this session to a close. May I ask you to join me to thank both Peter and Ka-shing for this wonderful panel discussion. Thank you.
Thank you. Thank you very much to our panel for this wonderful discussion. Please remain on stage. We'd like to have a photo of the three of you. We want together at the front of the stage. That was truly inspiring sharing from our three professors. During today's program, in the morning session at least, we heard several times that artificial intelligence was being brought up. The rise of AI is changing the way we live and work, but it's very important for us to be cautious about how we utilize it. Thank you so much once again, and thank you, Professor Chan, for moderating the session for us as well. Let's put our hands together once again to thank this panel for enlightening us. Thank you. The insights shared by our scientists this morning have shed light on the deep connections between life sciences and advancements in data and diagnostics. Thank you to our participants as well for your active participation. Let's carry forward the ideas shared today into your future conversations and collaborations. Thank you for joining us this morning.